All right, Bismillah. Let's do this. So, let's start with the development of the spinal cord and finish the subject. So, before I start, I just wanted to look at the physical, the spinal cord. What does it look like? Just to remind you about white matter, and about H-shaped gray matter. All right, and most importantly, you'll see that it has a canal in the middle, the Sumuha central canal, consists of CSF, and also lined by ependymal cells. All right, so I just wanted to show you what the spinal cord looks like, and especially the gray matter, which we have ventral dorsolateral horn. We wanted to see how is it developed. Okay, so let's start with the early development of the spinal cord. So let's translate this stuff real quick. So we know that the spinal cord is developed from where? From the neural tube. All right, we're going to start from here, from the neural tube. One third of it, as you know, gives you the brain, and 2/3 of it is going to give you the spinal cord. So we're going to focus on the spinal cord. Now, I wanted to do a cut section right here. Okay, I want to do a cut section or cross section of the neural tube, and I wanted to see superiorly what does it consist of. All right, so this is what it looks like superiorly. All right, you will see that it has a lumen, and I want you to believe me when I say that the lumen of the neural tube will eventually become the central canal of the spinal cord, which we've seen previously, and it will be consisting of just one layer. This one layer is سموها neuroepithelial layer, أو في اسم تاني سموها inner ventricular zone. Okay, so initially it's going to be composed of one layer, and this one layer is consisting of neuroepithelial cells. Now they are stem cells, تمام, that can differentiate into many different types of cells. The one different type of cell that it will differentiate into is يسموها neuroblasts. All right, يسموها هنا neuroblasts. And when they differentiate and proliferate into neuroblasts, they're going to form a new layer, a second layer. And this second layer is سموها هنا in the red color, the mantle layer, layer number 2. تمام? So you see neuroepithelial layer, you will see 3 layers eventually, but it's going to be composed of one layer, the mother. The mother will give birth to her child, the head of the child, the mantle layer, the neuroblast. تمام? And then the mantle layer, which is composed of the neuroblasts, they're just composed of cell bodies, all right? But eventually coming out of the person's head are his limbs. You see his limbs right here? Those are the nerve fibers of the neuroblasts. So the neuroblasts arising from them are the nerve fibers, and these nerve fibers, once they're created, they're going to form the third and outermost layer in the gray color. يسموها هنا marginal layer. تمام? And what does the marginal layer composed of? تمام? It is composed of the nerve fibers of the neuroblasts from the mantle layer. Okay, and where did the neuroblasts come from? From the neuroepithelial layer. Okay, so do you get an idea which of these layers are going to develop into what? Like, let's look at the last 2 layers. The mantle layer is going to give you the gray matter of the spinal cord. Wasn't it composed of neuroblasts? Neuroblasts are basically immature neurons. They could mature into sensory motor autonomic neurons. So they'll give you the H-shaped gray matter. What about the mantle layer? Didn't I say it's composed of nerve fibers? So what's it going to form? It's going to form the white matter of the spinal cord. All right, it's going to give you ascending, descending intersegmental fibers, right, of the spinal cord. Okay, so that's what we're going to form. One is going to form the future gray matter, and one's going to form the future white matter. Meanwhile, the neuroepithelial layer, is going to give you the neuroblasts, and some of it may also differentiate into ependymal cells and line the lumen of the neural tube, which I said we're going to become the central canal, right, and secrete CSF and so on. All right, that is, in summary, what you want to know from this slide, the early development that we've mentioned. And what we want to know from this slide as well, which 3 layers are going to give you which one is which. All right, perfect. So now we wanted to know in detail the last 2 layers, all right? The middle mantle layer and the outer marginal layer. How does it give you the gray matter? How does the mantle layer give you the future gray matter? And how does the marginal layer give you the future white matter of the spinal cord, all right? So let's look at the middle mantle layer. Now, what did I say the middle mantle layer is gonna be composed of? Neuroblasts, right? And these neuroblasts, من اسمهم, are immature neurons. They're going to differentiate into sensory neuron, motor neuron, and whatnot. All right. So let's look at this picture. So you see this circle? This circle is the same as this one before from this drawing. All right. The 3 layers. All right. It's the same. Just imagine I zoomed out like a microscope. I just zoomed out, and it's going to be like one small circle. All right. All right. It's the same. Now, next what happens, all right, is that coming out of it are going to be these bunny ears. You see these bunny ears right here? This is the bunny ears. And these bunny ears are coming out of the neural tube. All right, what are these bunny ears? These bunny ears are the neuroblasts of the mantle layer. All right, so basically, let me show you from previously. You see the mantle layer in the red color? Basically, they're going to proliferate from a dorsolateral direction. Which that dorsal direction, this is ventral direction, right? This is dorsal ventral direction. So it's going to proliferate dorsal lateral direction, and it's going to also proliferate ventral lateral direction. All right, and it's going to form these bunny ears. All right. It's going to form these bunny ears, right? As you can see. Now we give these bunny ears a name. We're going to call them as plates, all right? Now just remember, this is the ventral direction. We're looking from the ventral, and this is the dorsal direction, all right? Ventral, dorsal, so you don't get confused. The bunny ears from dorsally, we're going to give them a name. We're going to call it dorsal alar plate. And the bunny ears on the ventral side, we're going to call them ventral basal, I believe. We're going to check on the slide, but ventral basal plate, as is mentioned right here. All right? So you're going to have two plates. Now, just logic. What do you think the dorsal alar plate is going to give you specifically? Which part of the gray matter? It's going to give you the future dorsal horn. Remember the dorsal horn of the gray matter is going to give you the dorsal horn. And what is the dorsal horn composed of? Sensory neurons. So what about the ventral basal plate? What do you think it will develop into, mature into? It's going to give you the future ventral horn. And what does the ventral horn composed of, Nawaf? Composed of the motor neurons. All right? That's what these two plates are going to give you. These neuroblasts give you one sensory neuron here, motor neuron on the other side. All right? So what about the lateral horn? You remember the lateral horn found between T1 and L2 segments? We'll see. It's easy. We'll see it in another slide, so we don't get confused, all right? So this is all occurring where exactly? In which layer of the neural tube? This is on the marginal, not in the marginal, the mantle layer, all right? And this is all occurring in the middle mantle layer. So you see this white circle, all right? We finished the mantle layer, by the way. Do you see this white circle, حواليها? These white circle, this big white circle? This is your outer marginal layer, all right? This is going to be your outer marginal layer. And what did we say the outer marginal layer is going to be composed of? Nerve fibers, huh? So what do you think it's going to develop into? It's going to develop our outer marginal layer. It's going to develop into the white matter of the spinal cord. So it's going to develop into the three funiculus: dorsal, ventral, lateral funiculus, all right? So that's what this layer حواليها, this outer marginal layer, is going to develop into ascending, descending fibers to give you the white matter of the spinal cord. All right? Perfect. طيب, I want you to keep something in mind, all right? Is that as the neuroblasts of the middle mantle layer that you see right here are proliferating and make up these plates, right? As these plates are proliferating and bulging out, they're going to lead to some structural changes, all right? That's going to be seen in this picture, all right? I want you to tell me what are two changes you see immediately once the gray matter of the spinal cord is formed. I'll tell you two things. You see the lumen of the neural tube right here? You see how it got smaller, right? You see the comparison? They got smaller. Like I told you before, the lumen of the neural tube will become the central canal. So as the dorsal alar plate and the ventral basal plate, they bulge out, they proliferate outwards, they're going to make the lumen of the neural tube narrower or smaller, and they're going to form the central canal. Also, they will also make the formation of fissures, or septum, because the septum. You see how previously there was a big perfect circle? Well, now is going to be formation of two septum, my bad, two septum. One septum is going to be named after its location, tamam. Here, and don't get confused, this is not ventral side, all right? This is something from the other picture. But here in this septum, you see this septum right here? This septum is going to be known as dorso-medium septum, all right? Dorso-medium septum. And this septum right here, another septum ventrally, from this picture only, ventro-median septum, all right? And this is going to form your spinal cord completely, all right? Your spinal cord is finally going to be developed. Now, we have developed the gray matter of the spinal cord. The white matter also is developed, right? Consisting of ascending, descending fibers. The septa are formed, and the central canal is finally mature, right? Containing CSF, lined by ependymal cells. Now, let's talk about the lateral horn real quick, all right? The lateral horn is very easy. Let's look at this picture right here. This is the lateral horn. You see the alar plate right here, and you see the basal plate? You see the neurons in between them, right? These neurons here in between them, they want to proliferate, all right? But they have no space to go. They cannot proliferate medially ولا ventrally, ولا dorsally. So where they proliferate? These neurons are going to accumulate and they're going to form a horn on the lateral side. There's empty space here, right? So an accumulation, as is mentioned, of neurons between the alar and the basal plate will cause the formation of the lateral horn, or حتى يسمى intermediate horn. And what does this horn consist of? Motor neurons of the autonomic nervous system. يعني in T1L2 is going to be composed of sympathetic. في حتى هون كمان في الباراسمباثيك للاوتونوميك لكشر يسموها S2 between S2 S4 اللي هي parasympathetic horn. اوكي بس تعرف ان الهورن ده consists of autonomic motor neurons. اوكي between T1 و L2 للsympathetic, between S2 و S4 للباراسمباثيك. اوكي لازم تزودها. طب خلاص اهو ده السباينال كورد بالكامل، مش رسمها زي دي بالضبط، هي دي ده السباينال كورد بالكامل. تمام؟ ده structural changes I want you to keep in mind as the lateral plates and the ventral basal plates are proliferating. This is what I explained from this slide. Okay, that's nothing new. Perfect. So the spinal cord is officially developed. Next thing is myelination. Myelination, when does it occur first? When does it occur? During which months of pregnancy? During the 4th months of pregnancy. And here what it's trying to tell you is that the nerve fibers, all right, the nerve fibers in the spinal cord, they're going to be myelinated by oligodendroglial cells, right? So what about the nerve fibers outside the spinal cord, right? Outside the spinal cord, what are they going to be myelinated by? Schwann cells, all right? Easy. And if you want further clarification, it's here. This is the nerve fiber inside the spinal cord, myelinated by oligodendroglial cells. And outside the spinal cord is going to be myelinated instead by Schwann cells, all right? Perfect. We finished myelination, and this next slide is talking about the positional change of the spinal cord, right? Like this is the spinal cord, right? And this is the vertebrae. And as throughout development, as the baby develops more and more, you see initially his spinal cord was corresponding to his vertebrae, right? Completely. That's why they had coccygeal segment named after the fact that it was corresponding to the coccyx, right? But as he's born and he grows in length, thanks to his vertebral column increasing in length, the spinal cord will be at the level of L3, and in adulthood, it will be at the level of lower border of L1. This is all important for lumbar puncture purposes, okay? That's basically positional changes of the spinal cord, all right? Finally, we will go to the congenital anomalies of the spinal cord. It's simple. I'll just explain to you just one time, and inshallah, you will never forget it. Here is the structure of the spinal cord, right? Where would the spinal cord, the vertebrae class, this is the structure of the vertebrae. Where would the spinal cord be? Here, in the vertebral foramen, right? And the spinal cord will be locked in here. It will not bulge out, all right? It will not bulge out. None of its content will bulge out. Because we have here what is called as a spinous process. How is the spinous process formed? By the fusion of the vertebral arches. Vertebral arches عبارة عن pedicle وlamina بيعملوا fusion عشان يعطيك spinous process. And this is again from posterior view. And if you bend forward, you can feel in a skinny person his spinous process. So this spinous process is meant to keep the spinal cord, all right, from being bulging out. تمام? Keep it in the vertebrae. So what happens if there is failure of fusion? What happens if the vertebral arches, they don't fuse to give you the spinous process? Like in this picture right here. What happens? Well, this will lead to an opening right here. And this opening, if large enough, will cause the bulging of the spinal cord, its contents, or its spinal cord itself, or its meninges, or CSF. They could bulge out right here, and they could form a sac, depending on the type. Or maybe they don't bulge out. Again, like I said, if the opening is small. So if there's failure of fusion of these vertebral arches, this could lead to an opening. Like you see this opening right here. And if it's large enough, there could be bulging of the spina bifida. But if this opening is small, this could lead to a type of spina bifida where he has spina bifida, but it doesn't bulge out. That type is spina bifida oculata, okay? But let me actually explain to you just quickly why is there failure of fusion of the spinal cord? Of the vertebral arches. Why is there failure of fusion of the vertebral arches? All right? So spine يسموها spine or vertebral arches. Bifida معناها split, okay? Let's see why. All right. Let me remind you the the 3 discs or the 3 layers of the embryo, right? This is the ectoderm. There's 3 layers: the ectoderm, mesoderm, endoderm. This is the neural tube, all right? And you see the neural tube, once it's formed, it's going to be swimming in the mesoderm. This is important, all right? Now, I want you to keep in mind, the CNS is going to come from the neural tube. The vertebrae, the formation of the vertebrae, all these parts is going to be formed from the mesoderm, all right? It's going to form all from the mesoderm. So what I want you to keep in mind is look at this drawing right here. This is the neural tube. You see the neural tube right here? You see, the neural tube, as we know, should be closed completely, right? It should be closed completely, and for two reasons. One, the neural tube, the development of the CNS, must be separate from the development of the skin from the surface ectoderm. Because what would happen, for example, if there was an opening of the neural tube, right, on the caudal end? Like, for example, if I were to draw it right here, right? There's an opening, a gap right here. What would happen? This would lead to adhesion بين النeural tube وما بين الsurface ectoderm. هنشوفها بعدين. This is another reason for the last anomaly, spinal dermal sinus. This is one of the reasons it should be closed. Another reason it should be closed is for this lovely mesoderm. I told you the mesoderm is going to give you the vertebrae, right? It's going to give you the vertebrae, all of it. You see, the mesoderm wants the neural tube to be closed so it can hug it and form the vertebra around the neural tube. So if the neural tube is closed, it can form the entire vertebra, the body, the vertebral arches, as long as the neural tube is closed. It can hug it, right? Hug around it, essentially. So that the neural tube is inside the vertebral column, as you can see. The spinal cord will be inside the vertebral column, and the skin will be outside. Right here, okay? Now, if there was an opening, then the mesodermal cells, they cannot close the gap, all right? The mesodermal cells, they cannot hug this gap. And in that case, part of the mesoderm responsible for forming the vertebral arches and fuse them together to give you the spinous process, khalas, it will not be there. Thus, there will be split spine. There will be what, Nawaf? Spina bifida. All right, there will be spina bifida. Because there is failure, in short, in summary, failure of closure of the caudal end of the neural tube. Okay, failure of closure of the neural tube. Okay, that's why there's non-fusion. Khalas? Perfect. This is the reason for all types of spina bifida. There's 4 types. All of them have the same reason. The only difference between the 4 types of spina bifida we're about to see is that the difference is whether the spinal cord, will it bulge out, only the spinal cord, يسمى myelocele, or will it be the meninges, right? Or will it be both spinal cord and meninges, or will it not protrude at all? Well, the spinal cord and its contents never protrude at all. يسمى spina bifida oculata, okay? خلينا نشوف الفور تايبس، اوكي؟ جست نشوف الاختلاف البروتروجن، ايش الكونتنتس، تمام؟ حلو، خلصنا ده أصعب شيء وهسه أسهل شيء بجد. سباينا بيفيدا اوكيلاتا. اوكيلاتا معناها هيدن، اوكي؟ معناها هيدن. ليش هيدن؟ معناها عنده الشخص البيبي عنده سباينا بيفيدا بس he does not have any clinical symptoms. Why? حصل spina bifida, but you can see the opening is small. It's small enough that the contents does not bulge out. بس حصل له spina bifida. So he will not be suffering of any clinical symptoms, but there is an evidence you could confirm that this is spina bifida oculata. You can diagnose it, but the fact that there's a small dimple معناها depression, with a tuft of hair. So يجالك MCQ, a baby with small dimple with tuft of hair on his back, L5 or S5, تمام? Then this is spina bifida oculata, okay? ما فيش أي bulging ولا شيء. زي ما شفنا, a dimple with some tufts of hair. اللي بعده spina bifida meningocele. Meningio معناها meninges. Seal معناها cyst. So, what is the features of spina bifida meningocele? إنه في cyst-like sac in his back, right? L5 or S1. So, what does the cyst-like sac consist of? It consists of the meninges اللي حواليها, and it consists of cerebrospinal fluid. تمام? But what about the spinal cord? أنا قلت لك meningocele. Is the spinal cord bulging out as well? No, the spinal cord and the spinal roots are intact. Just the cerebrospinal fluid and the meninges are bulging out to give you this cyst-like sac. وطبعا ده هتؤدي إلى neurological deficit. ليش؟ لأنه it's in his back. Imagine the baby during pregnancy. His back is being compressed against the wall of the uterus. So this means if there's a sac, it can compress. It can lead to neurological deficit because there's compression against the wall of the uterus during pregnancy, right? Because it is sac. So if there's neurological deficit, and it can lead to paralysis. Okay, it can lead to paralysis. So the spina bifida meningocele. زي ما شفنا هنا. اللي بعده spina bifida meningomyelocele. نفس الشيء بس خد كلمة مايلو معناها spinal cord, okay? Spinal cord. نفس الشيء بيكون عنده severe neurological deficit, صح? Because the spinal cord is involved, طبعا. So this is spina bifida meningomyelocele. وآخر شيء معانا اللي هي spina bifida myelocele. Is the meninges bulging out? No. CSF bulging out? No. فقط spinal cord. وتلاحظ in this picture right here. You see, this is the meninges, right? This pink color. And this one حواليه ما المننجز قصدي. This is the CSF. And this is the meninges, right? The CSF meninges underneath. And what's bulging out is only the spinal cord. طب you see a difference? You see something noticeable about the spina bifida myelocele? You see, there's no skin. You see, the spinal cord is exposed to the surface. There's no development of skin. I don't have to tell you the reason. You don't have to know, but you just have to know that the spinal bifida myelocele, there is no formation of skin. The spinal cord here is actually exposed to the surface. So there's no formation of a cyst-like sac, the meninge is not protruding, right, with CSF, and there's no formation of skin. So the spinal cord is exposed to the surface. Why you should know this? Because of this, all right? Look at this. This is a guaranteed MCQ. It says here that for diagnosis of spina bifida myelocele, there's going to be high level of alpha-fetoprotein in the amniotic fluid. Why? Well, let me draw here the amniotic fluid, all right? Because this is from the picture of the baby from the back, all right? And here from outside the skin, you have the amniotic fluid during pregnancy. You see, if there is no formation of skin and the spinal cord is exposed to the surface, you remember the alpha-fetoprotein that's developed from the baby's liver? It could leak out. It could leak out to the CSF, and then it could leak out to outside the skin because there's no formation of skin. It can leak out of the spinal cord and it can accumulate in the amniotic fluid, all right? So if you could do an antenatal follow-up or a prenatal follow-up, you do amniocentesis from the mother, right? And you look at the amniotic fluid and you saw high levels of alpha-fetoprotein, this is your diagnosis of spina bifida myelocele, okay? This is MCQ guaranteed, tamam? Besides that, it has a severe neurological deficit as, the same thing, okay? Myelocele. But this is very important what I just mentioned about high alpha-fetoprotein level, okay? The last anomaly of the spinal cord is the spinal dermal sinus. Spinal معناها spine, spinal cord. Dermal معناها skin. Sinus, what does it mean? يعني معناها channel، يعني معناها كده channel. A narrow channel زي كده. زي الصورة دي اللي هنشوفها، طيب؟ Now, spinal dermal sinus, I'll just explain it once and inshallah you'll never get it. You'll get the reason why it's called spinal dermal sinus. The cause is telling you there's failure of closure of the caudal neuropore at the end of the 4th week. نفس السبب اللي قلت لك من قبل, okay? اللي قلت لك إنه neural tube, it must be closed, right? For the mesoderm to hug it, right? For tube reformation. And also the neural tube must be closed so the development of the CNS is separate from the development of the skin, surface ectoderm. Surface ectoderm separate from the neuroectoderm. لو في gap لما يشوفنا هنا زي النيورال تيوب دي, لو حصل في gap هيكون في adhesion مع بين السكن وادهيجن مع بين السباينال كورد, تمام؟ يعني السيرفس مع النيورو اكتوديرم. واذا حصل adhesion there will be formation of a sinus, this sinus right here, extending from the spine from the inside. This sinus extending from the spine all the way to the skin from the outside. And this is what you would see MRI lateral view. You don't have to know it, but just for explanation. So what is the feature of the spinal dermal sinus from the outside, right? What is the feature of it from the tract here in the skin? This is what it would look like from the outside, right? This is the tract from the outside. You'll see a formation, not formation, but the presence of a hyperpigmented dimple right here in the back, in the sacral region, here as well. And if you did a sagittal view, this is what you would see. You would see a tract or a sinus ما بين الspine والskin. لأنه ليش؟ لأنه في failure of closure of the caudal neuropore. بيحصل adhesion ما بين النيوروectoderm بيعطيك الspinal cord والsurface ectoderm اللي بيعطيك الskin. Okay, spinal dermal sinus. بس تعرف الكوزز والفيتشر بس للفهم, okay? فبس ده للspinal dermal sinus وخلصنا الليكتشر بتاعت الspinal cord. شكرا جزيلا, مع السلامة, good luck, رمضان كريم or عيد الفطر or whatever.